So today I'm going to be talking about mutations and how they work and just some examples of mutations. So to start off, mutations are anything that are any permanent changes in the base sequences of DNA. So like the nucleotides, the codons, etc. within DNA. Any permanent changes in those are considered mutations. So for example, if you were to mutate one of the nucleotides on this 3-sequence codon, so if we change cytosine to guanine, that would be considered a mutation. This would be later present in transcription, translation. An important term to know are mutagens. Mutagens are agents that cause mutations. That's anything that is actually causing the mutation itself. And I can talk about a few of those agents in a little bit, but mutagens are agents that cause mutations. Now, mutations aren't necessarily bad all the time. They could be neutral, meaning that there's no real effect of the mutation. There's beneficial mutations and there are harmful mutations. An example of a neutral mutation is anything like in the non-coding region of DNA. Usually that has no effect on gene expression or protein function. An example of this is glutamic acid. So the structure of the 3-sequence codon that codes for glutamic acid is guanine, adenine, and guanine. Guanine, adenine, and adenine. A common mutation of that is guanine, adenine, and then the adenine getting replaced for guanine, so GAG. So even though that 3-sequence codon changes, the codon still codes for it's still the same amino acid, so still the same function and everything, so everything stays the same, so there's not going to be a real problem with that. Therefore it's neutral. A beneficial mutation is an example of bacteria gaining antibiotic resistance, so that is considered a mutation. This is very beneficial obviously for bacteria. In humans, the deletion of the CCR5 gene is a mutation, and essentially that will make people more resistant to HIV. Harmful mutations could be anything from skin cancer, from UV radiation, sickle cell anemia, which is a point mutation, which I'll talk about in a second, cystic fibrosis. Those are all examples of harmful mutations. So a point mutation is anything that changes a single base pair, kind of like the example I gave up top. So if you're just changing in this codon, in this 3-sequence codon of ATC, if you were to change the cytosine to a guanine, that would be considered a point mutation. There are kind of 3 branches of a point mutation. There's silent mutations, so similar to the neutral example I gave, there would be no change in the amino acid, so that's kind of neutral. A missense mutation is if you were to change the nucleotide in this codon and it codes for a different amino acid, that would be considered a missense mutation, so that could affect the protein the protein actually being synthesized. So a missense is essentially it creates a different amino acid than what we wanted originally. A nonsense mutation is anything that will turn a normal codon into a stop codon. There are also frame shift mutations. Frame shift mutations they kind of go hand in hand with another kind of mutation, or I'll get into that in a second. It's a little hard for me to explain right now. So a frame shift mutation is when one or more nucleotides gets added to a 3-sequence codon. Because codons are read in groups of 3, adding or deleting bases, nucleotides, will change how every codon after that point is read. So for example, if we have a repeat of ATC, let me just write it down. If we have a repeat of ATC, right? And we insert an extra nucleotide, so we say we insert a C. It was then being read from ATC, but then we add the extra cytosine, so it goes ATC, then it goes to CAT, and then so on, right? So then what we started with was ATC, and it transferred to CAT. So that is considered a frame shift mutation. So a frame, so like let's think of a frame right here. So codons are read in groups of 3. It's just changing how every codon after that is read. So that is considered a frame shift mutation. This could also fall under the category of missense and nonsense mutations. The other kind of mutation that goes hand in hand with frame shift mutations are insertion and deletion mutations. So again, that's when one or more nucleotides gets added or lost. Frame shift mutations are a consequence of this only if the number of nucleotides inserted or deleted is not in multiples of 3. So if it's less than 3, then a frame shift mutation could occur. If it's more than 3, then it wouldn't be a frame shift mutation. So in terms of mutagens are kind of broken up into chemical mutagens and physical mutagens. Chemical mutagens are a little bit more tricky to learn and to kind of explain, at least for me. A mutagen, again, is an agent that will cause a mutation. So base analogs is a type of mutagen. There are chemicals that will resemble normal DNA bases, so normal nucleotides, whatever, so closely that the cell mistakes them for nucleotides. So an example of a fake nucleotide or base analog would be 5-bromouracil. 5-bromouracil looks like thymine. 5B will look like thymine, and it will actually pair to adenine sometimes. So that's kind of an example of a base analog. Base modifying agents are agents that will modify the chemical structure of existing DNA bases. So this will ultimately change how they pair during replication. Nitrous acid, for example, or nitrous acid is an example of a base modifying agent. This will actually cause adenine to bind to cytosine instead of thymine. So this would actually lead to a point mutation. So again, base modifying agents, anything that will chemically alter the structure of existing DNA bases. So intercalating agents are anything that will insert themselves in between base pairs. So this will ultimately lead to a frameshift mutation, just because if you do insert, or if you just add something to like a 3-sequence codon or whatever, if you just add one of them in there, that will definitely lead to a frameshift mutation. So we also have physical mutagens. So physical mutagens are a little bit simpler, or I'm sure what people are more familiar with. We have ionizing radiation, so those are X-rays, gamma rays, etc. And we also have non-ionizing radiation, so that's UV light.